Ouch.
What's wrong?
I just cut myself.
Oh, I'm so sorry, man.
What do I do?
Not you. What do your cells do?
What do you mean?
What I mean is mitosis. And that's how you cure cancer. Oh hey, I didn't see you there. I presume you're here for mitosis? Well, let's get right to it. Well, this is the cell cycle. It consists of interphase and cell division, also called mitosis. Interphase has three main stages: the G phase, the S phase, and the G2 phase. In G1, or the first growth phase, the cell grows and makes new organelles and proteins. In synthesis phase, the centrioles and chromatin are duplicated. In G2, which is the second growth phase, enzymes to help with cell division are made. Now that you know about interphase, we can get right to mitosis. As you can see, cell division is actually a small stage of the whole cell cycle. Mitosis consists of prophase, metaphase, anaphase, telophase, and cytokinesis.
In prophase, the chromatin condenses into chromosomes and turn visible. The chromosomes are two sister chromatids that are banded together by a centromere. Both centrioles grow a spindle system and start to travel towards opposite poles. The nuclear envelope then breaks down until there is no nucleus. The spindle fibers from each pole attach to the centromere of the chromosomes.
In metaphase, the spindles line up the chromosomes in a straight line in the center of the cell.
In anaphase, the spindle pulls the sister chromatids away, one heading to one pole and the other to the opposite pole. Now, the chromatids are individual.
In telophase, the spindles break down now that the chromosomes are at opposite poles. They group up with the other chromosomes at their side to form their own nuclei. Now, there are two nuclei at both opposite ends. Cytokinesis ends mitosis by extending the cells further to completely separate the two cells.
Okay, so now I know about animal and human mitosis, but what about plant mitosis? Is it the same?
It's similar, but not exactly the same. During the course of interphase, the chromosomes replicate, turning into two sister chromatids.
Prophase. During the prophase, genetic material called chromatin, which contains DNA and proteins, condenses to form chromosomes. The chromosomes then turns into two identical copies of itself that are joined together at a central point called the centromere. Then, structures called centrosome develop on the outside of the nucleus. The centrosome creates hollow rods called microtubules that will enter the nucleus to attach itself to the chromosomes at their centromeres.
In metaphase, every chromosome is completely condensed. The centromeres and the microtubules move to different ends of the cell. This causes the chromosomes to pull each other towards opposite ends of the cell. The centromeres also start to form a point of separation during this stage.
Anaphase. When a plant cell is in anaphase, the sister chromatids separate into two pairs. One half moves to one end of the cell, while the other half moves to the other end. Microtubules in the cell also work to lengthen the plant cell in order to prepare it for telophase.
In the telophase stage, the cell divides the cytoplasm into two portions. The microtubules and the centrosomes disintegrate. The rest of the nucleus from the original cell creates a nucleus at the two opposite sides of the cell around the chromosomes. In this phase, the chromosomes turn into chromatin once more.
Cytokinesis. When a plant cell enters cytokinesis, it makes a cell plate which will be used to divide the cell into two separate daughter cells. And finally, the two daughter cells pull apart from each other to become individual cells with an exact copy of the original cell's genetic material.